mais lui, il y a une approche aussi qui est une approche très intéressante sur l'origine des virus. So, you're welcome, and we'll be happy to hear you.
Thank you for the introduction. Hello, everyone. And first of all, thank you for inviting me to give this seminar, and it's a pleasure to be in Marseille. And today I will talk about evolutionary relationships between different selfish elements, including viruses, plasmids, and transposons. So this is one way to depict the virusphere, and the punchline is that viruses are extremely diverse. So they can come in a variety of shapes and sizes, and they can have different nucleic acids as the genetic material: RNA, DNA, double-stranded, single-stranded, linear, circular, segmented, or monopartite. So, and all combinations of these. And on top of that, there are plenty of evolutionary relationships between all these viruses and different other types of mobile genetic elements, such as plasmids and transposons. And my interest is to understand how different groups within this huge virusphere relate to each other, and what are the evolutionary connections between different viruses, and also between viruses and other types of mobile genetic elements. So plasmids and transposons. So it includes everything. Okay, and in my talk I will speak about these connections between different mobile genetic elements on two different systems. So for the most part of my talk I will speak about viruses with single-stranded DNA genomes. And these are among the smallest viruses known. And then in the second part, which will be shorter, I will also come to viruses which are on the other side of the size spectrum. And these are viruses with double-stranded DNA genomes. Okay, so first about the single-stranded DNA viruses. So these viruses infect hosts in all three domains of life, and different domains have associated kind of specific groups of single-stranded DNA viruses. And these viruses are highly abundant in the environment, so they are present in all possible environments you can think of, from lakes to human gut to boiling springs and so on. And in my talk I will speak specifically about viruses infecting eukaryotes, and not touch upon the archaeal or bacterial ones. So all eukaryotic viruses with single-stranded DNA genomes are quite uniform in terms of morphology. So all of them have small icosahedral capsids, which can be either isometric or geminid. And these small capsids can accommodate only a small piece of genetic information, and the genomes of these viruses do not usually reach 10 kb. And in comparison to prokaryotic viruses, viruses of eukaryotes have smaller genomes, and prokaryotes can have a little bit bigger ones. And we have recently isolated a spiral-shaped virus from archaea, hyperthermophilic archaea, which had 25 kb, and that was like a mini virus among all single-stranded DNA viruses, so we were very happy. But let's get back to eukaryotes. So eukaryotic viruses, as I said, are very small, and the genomes are also very simple. So many of them have just two genes, like this circovirus, for example. One gene encodes for the capsid protein, and the other one for the replication protein. And this is just one step above of being a plasmid. And some plasmids indeed encode only one gene for the replication protein. So this kind of irreducible simplicity of single-stranded DNA viruses makes them a very good model to study the origin and evolution of viruses. And that is what I'm interested in. So in principle, you can think that you try to figure out where different genes in the viral genome come from, and this will help you to understand the origin of different specific virus groups. So when you look at these genes, the answer is not immediately obvious. And single-stranded DNA viruses, they display a specific bilateral evolutionary relationship. So on the one hand, they are related to rolling circle plasmids, which are abundant in bacteria. But on the other hand, structurally they are very much similar to viruses with RNA genomes, which in fact you carry. So what does this mean, and how these two types of elements, RNA viruses and plasmids, relate to the evolution of single-stranded DNA viruses? So that's what we try to figure out. So the vast majority of eukaryotic single-stranded DNA viruses replicate by the rolling circle mechanism or adaptation of this mechanism, which is mediated by virus-encoded endonuclease protein. And this endonuclease nicks the double-stranded replicative intermediate form, and when 3' OH is released, which is then extended by the host DNA polymerase to produce a new double-stranded DNA molecule, and exposing single-stranded genomic DNA of the virus. And when you look at these endonuclease proteins in all eukaryotic viruses, all eukaryotic single-stranded DNA viruses which we know of, this protein consists of two different domains. So the N-terminal domain has the endonuclease domain, endonuclease activity, which is involved in nicking the genomic DNA, and then at the C-terminus there is superfamily free helicase domain. And this kind of two-domain organization is not found in any of the bacterial or archaeal single-stranded DNA viruses. So it doesn't seem that eukaryotic viruses came directly from prokaryotic ones. Instead, many bacterial plasmids encode this specific domain organization with the N-terminal endonuclease domain and the C-terminal superfamily free helicase domain. So this implies that there is some kind of evolutionary link between these bacterial plasmids and single-stranded DNA viruses of eukaryotes. On the other hand, all structures of capsid proteins from single-stranded DNA viruses have one specific topology, and which is known as the jelly roll fold, which is depicted here schematically, which looks like a jelly roll cake. So that's where the name of the fold comes from. And this particular topology of the capsid protein is also found in the vast majority of icosahedral single-stranded RNA viruses. And the list you can see here in green is extensive and includes many different families and unclassified genera of single-stranded RNA viruses. So, and very often you hear claims that structural similarity between viral capsids might mean nothing because proteins converge to a specific topology. However, given our current understanding from the, and this convergence argument is based on the fact that, I don't know, about, it's based on the negligence probably more than on some real evidence, because given our understanding of the diversity and structures of different viral capsid proteins which can form icosahedral capsids, which some of which are shown here on this slide, you can see that icosahedra can be built from proteins with exclusively alpha helical folds to proteins which are dominated by beta strands. And here on the right you have two examples of two icosahedral virions. So one at the top is constructed from exclusively alpha helical protein, and then at the bottom the one which is built from the beta strand-rich capsid protein. So there is no particular reason why this alpha helical protein should evolve to something else than it is now. So there is no reason why capsid proteins would have to converge to one particular topology. Any kind of fold, if it's suited to be icosahedron, it will build it. And so what I want to say is that structural similarity between the capsid proteins in single-stranded DNA viruses and single-stranded RNA viruses must mean some kind of evolutionary link between these different groups of viruses. And along these lines, a few years ago, we were investigating the origin and evolution of plant-infecting viruses of the Geminiviridae family. So Gemini viruses infect plants, and here on the background of the slide you can see the virion structure of this virus. So it is built from two incomplete icosahedra, which are glued together to produce these geminid particles. And when we looked at the capsid protein of Gemini viruses, so from different genera of Gemini viridae, they all have the same kind of fold, which is a jelly roll fold. And this jelly roll fold is closely related to the fold of the capsid protein from satellite tobacco necrosis virus. And satellite tobacco necrosis virus, it also infects plants as Gemini viruses do, but it has a single-stranded RNA genome. We next looked at the replication protein of Gemini viruses, and what we found is that the Gemini viral replication proteins are most closely related to the replication proteins from phytoplasmas. And phytoplasmas are obligate intracellular parasites of plants, so they replicate exactly where Gemini viruses live. And this relationship between phytoplasmal plasmids and Gemini viruses was to the exclusion of all other groups of eukaryotic single-stranded DNA viruses, such as circoviruses or nanoviruses. So this implied that there is some kind of link between these bacterial plasmids and Gemini viruses. And a priori, this kind of relationship can be explained by two opposite possibilities. So first one is that phytoplasmal plasmids are Gemini viruses which learn how to replicate in bacteria. And the opposite is that Gemini viruses are in fact bacterial plasmids which learn how to replicate in the cytoplasm. And there is support for the latter hypothesis that specifically that Gemini viruses are kind of derived from bacterial replicants. And in fact, if you analyze the genomes of Gemini viruses, you find that they still have bacterial promoters. And these promoters, if they are used in bacteria under some reporter genes, they are still active in bacteria from Gemini viruses. And they are active both in E. coli and in agrobacterium. And in fact, if you introduce the Gemini virus genome into a bacterial cell, it will replicate there in a replication protein-dependent manner. So this indeed indicates that Gemini viruses are derivatives of bacterial plasmids. So considering this evidence on the structural protein and the replication protein, we came up with the following scenario for the origin of Gemini viruses, in which Gemini viruses are originating from bacterial phytoplasmal plasmids upon acquisition of a capsid encoding gene from an RNA virus. And the weak point of this hypothesis is that it involves recombination between two replicants, one a DNA replicant and the other RNA replicant. And the evidence for this kind of unusual recombination was not so prevalent at the time. However, more recently, a new group of viruses, which we call chimeric viruses, has been discovered. And in 2012, Ken Stedman and Jeffrey Demer reported a new genome, viral genome from some lake sample, and they analyzed the genome, and what they found is that the replication protein of this viral virus had a close relationship to the replication protein from single-stranded DNA viruses of the Circoviridae family. However, when they looked at the capsid protein gene, there was nothing to be similar to be found in this capsid protein to the circoviral capsid protein. Instead, it was closely related to the capsid proteins, specific topology of the capsid proteins found in single-stranded RNA viruses of the Tombus-Viridae family. So this gene pattern is most consistent with a recombination between a single-stranded DNA virus, a single-stranded RNA virus, which then would give rise to this chimeric entity where two genes are coming from different sources. So, and if you look at this chimeric entity, so in its appearance and size, it would be practically indistinguishable from the RNA virus. But when you open it up and you look inside, So you will find the genetic material which is then DNA instead of RNA, and which would be replicated like in the case of these single-stranded DNA viruses. So this was exciting and puzzling discovery, and we wanted to learn more about these viruses, and for this we have looked in all available virals and assembled all contigs which would have like a capsid protein gene from RNA viruses and a replication protein from DNA virus. And what we found is 13 new genomes with this kind of organization, and these were from different aquatic environments, freshwater and marine. And although we were not targeting one specific capsid protein, all of these 13 guys, we had this Tombus virus-like capsid protein as found by Diemer and Stedman. And this allowed us to build some meaningful tree, and what it showed is that all capsid proteins from chimeric viruses, we formed a monophyletic group coming from within the RNA virus capsid protein, suggesting that there was a single acquisition event of the capsid protein gene by the ancestor of all this group of chimeric viruses. Then we looked at the replication protein. And typically, when you analyze these rolling circle replication proteins of single-stranded DNA viruses in eukaryotes, viruses from different families, we form well-supported clusters, and then they don't mix genes, they don't exchange genes. So, for example, Gemini viruses always come together when nanoviruses form a group of their own, and circoviruses as well. And then we threw in our chimeric virus replication protein. And all these, they were scattered all over the place. So some were grouping with Gemini viruses, others with nanoviruses, and a third group with circoviruses. So indicating that there was a turbulent kind of evolution in the group of chimeric viruses. And this is most compatible with the following scenario, that there was a recombination event with, between DNA virus and RNA virus producing this chimeric virus ancestor, and then within this ancestor experienced multiple recurrent exchanges of different replication proteins with viruses which are distantly related from different families. And more recently, we fished out another chimeric virus, and this time it did not come from any environmental sample, but instead it was a contamination of a QIAGEN kit. So we didn't care about that, so or we were lucky to have it, this contaminated kit. And the capsid protein again was of the same variety, Tombus virus-like, nothing particular, grouped with all the other chimeric viruses. And then we looked at the replication protein of this virus. Again, if you have these three clades of different families, circoviruses, nanoviruses, and Gemini viruses, and the chimeric viruses grouped wherever they want. And our new virus, which is called CHIP14, was somewhat in between the nanoviruses and Gemini viruses, which was kind of not very right. So, as I mentioned, the replication proteins of these viruses, they consist of two domains. So the N-terminal part encodes the endonuclease, and the C-terminal part encodes the helicase. So we decided to look at the domains separately, and what we found is that when only N-terminal domain was considered, our chimeric new virus, it jumped straight into the group of Gemini viruses, branching right where like a Gemini virus protein. But then when we looked at the C-terminal part of the protein, instead of grouping with Gemini viruses, it firmly clustered with nanoviruses. So what this means is that these chimeric viruses, they are not only chimeras at the level of genes, but also the domains within these genes are also chimeric, coming from different families of viruses. So they are ultimate chimeras. And this is quite different from all other known single-stranded DNA viruses, because like on these trees you can see that circoviruses either circoviruses, nanoviruses, or Gemini viruses, in all the trees they are forming uniform plates, and they do not exchange neither genes nor the domains between themselves. So chimeric viruses are really unique in this perspective. So, why we care about this is that because this substantiates the hypothesis on the origin of single-stranded DNA viruses from bacterial plasmids. So this exchange of or recombination between DNA replicons and RNA viruses, and specifically transfer of capsid encoding genes between widely different entities is possible, and we have proof of that. So this means that that these single-stranded DNA viruses and single-stranded RNA viruses and perhaps plasmids, they should be considered in the framework of evolutionary continuum rather than as some completely different entities which are placed on different parts of the virusphere and should not even be talked about in the same presentation. Instead, it is all kind of coming together in one continuous thing. And once in existence, these single-stranded DNA viruses, of course, they diversify, and they follow different evolutionary paths, and sometimes they take very extreme paths. And I will give you one example in the next few slides about the extreme genetic mosaicism in the family Bidnaviridae. So Bidna viruses, in fact, silkworm Bombyx mori, and they have bipartite, linear, single-stranded DNA genomes with terminal inverted repeats. And these features, linear genomes with terminal repeats and icosahedral capsids, is similar to viruses of the Parvoviridae family. And initially, Bidna viruses were indeed classified within the Parvoviridae group. However, unlike parvoviruses, Bidna viruses do not encode the endonuclease, this rolling circle endonuclease protein which is nicking the genome. Instead, uniquely, these viruses encode protein prime family B DNA polymerase, and also the typical parvovirus capsid protein with the jelly roll fold could not be initially identified in Bidna viruses. So due to these differences, Bidna viruses were exempted from this Parvoviridae family and placed into the family Bidnaviridae instead. So with Eugene Koonin, we set out to understand the evolutionary history of this interesting virus group, and for that we systematically analyzed the evolution or origin of the different genes within Bidna virus. And what we found is that we could find that Bidna viruses are indeed genuine descendants of parvoviruses. So we identified a homologue of the jelly roll capsid protein within the Bidna virus, and also, although Bidna viruses do not encode the endonuclease, they still have a remnant of this replication protein of parvoviruses, but only the C-terminal part of it, the helicase domain. So they still have it, and it groups within the helicases of these parvoviruses infecting insects. So, and then the most kind of radical change in the history of Bidna viruses was obviously loss of the endonuclease domain and acquisition of the family B DNA polymerase by these viruses. And the analysis of this polymerase gene showed that it was placed firmly within the group of large DNA transporters, which are known as polyntops. So Bidna viruses acquired the polymerase gene from eukaryotic DNA transposons. And then we looked at the second segment of the Bidna virus genome, and then we found that one of the genes was in fact acquired from large DNA viruses, double-stranded DNA viruses of the Baculoviridae family. And the other gene was acquired from viruses with double-stranded RNA genome from the Reoviridae family. So this is a really mosaic of different genes coming from different sources. So in fact, BIDNA viruses are a result of gene shuffling between 4 groups of selfish genetic elements with single-stranded DNA genomes, double-stranded DNA genomes, double-stranded RNA genomes, and some DNA transposons. And how this could have happened, we came up with the following scenario to explain this gene shuffling. So, according to our thinking, parvovirus, ancestor of BIDNA viruses, has properly integrated into the genome of polyton transposon. And parvoviruses are indeed known to integrate using the endonuclease activity into the genomes of their host, and then subsequent loss of most of the genes from the polyton and the endonuclease domain from the ancestral parvovirus would produce a genome of monopartite BIDNA virus, and then exchange and acquisition of genes from baculoviruses and reoviruses would produce the gene complement what we have now in BIDNA viruses. Another important thing to mention about single-stranded DNA viruses and for their evolution is that single-stranded DNA viruses have the inherent ability to integrate into the genomes of their host. And for this, eukaryotic members of this virus class, they do not require any host genes or any additional genes, and instead they use the endonuclease activity of the replication protein. And this has been shown very well for parvoviruses and also with linear genomes, and also the single-stranded viruses with circular genomes can also integrate using the activity of the proteins. And this is important because integration into different other DNA molecules of different sources, viral genomes or host genomes, this provides viruses with the opportunity to exchange genes and form very different new recombinant entities. And for example, in some organisms, like in this Sacoglossus kovalevsky genome, there are 50 copies of parvovirus-like elements which include both the capsid protein and the replication proteins. And some of these single-stranded DNA viruses seem to have abandoned completely the virus kind of lifestyle and resumed as genuine transposons. For example, in the Hydra magnipoculata, there are 400 copies of elements which encode parvovirus-like replication protein, but no capsid protein. So it seems that in this case, there was a shift from being a virus to completely being a transposon. That doesn't exclude the possibility that this element now abundant within the genome of the will eventually acquire capsid protein from somewhere else and spring out as a virus at some point of its life. And here on this slide I put some evolutionary transitions between different elements spinning around the viruses with single-stranded DNA viruses. So in all likelihood, this group of single-stranded DNA viruses has been molded by genes from widely different sources. So specifically, the replication protein probably came from rolling circle plasmids, whereas the capsid protein genes were acquired from RNA viruses. And once in existence, these viruses, of course, diversified. So in some instances, the replication protein was exchanged for something completely different, specifically for DNA polymerase. And this is a big transition, being able to synthesize your own DNA instead of using the host polymerase. Other viruses, like these chimeric viruses, they exchange the capsid protein. So all the components of the genome are in fact exchangeable and have been exchanged in the history of this virus group. And there is also evidence that single-stranded DNA viruses gave rise to certain groups of double-stranded DNA viruses, specifically of polyoma and papillomaviridae, and some groups of rolling circle transposons have also emerged directly from single-stranded DNA viruses. So all these selfish elements with very different genomes, RNA, DNA, single-stranded, double-stranded, plasmids, viruses, transposons. So they all are in the same mix, and when they can, they do exchange genes, and they generate this diversity within the virus which we observe. Okay, and they should be probably considered within the framework of continuum rather than be a kind of segments of their own within this global virusphere and mobilome. So in the next part of my talk, and this part will be shorter than the previous one, I will talk about our recent work on DNA transposons of a Polyton superfamily. And I briefly mentioned them in relation to the evolution of single-stranded BIDNA viruses, but now I will talk about the contribution to the evolution of some groups of double-stranded DNA viruses. So what are the polyntons? So polyntons, also known as mavericks, they have been discovered some 10 years ago by two groups independently and almost simultaneously. And these are big transposons of eukaryotes, and they are big according to the standards of transposons. So they can be around 15-20 kb in size, and they encode retrovirus-like integrase and family B DNA polymerase, which is protein prime, and this is actually where the name Polyton comes from. Pol stands for polymerase and int stands for integrase. And these elements are widely distributed in eukaryotes. So we are present in all kinds of organisms, from multicellular to unicellular, from amoeba to alligator. So, and this suggests that the origin within eukarya is ancient, and that these elements seem to be very successful. Okay, so in addition to the polymerase and integrase gene, a recent study has revealed that these polyntons encode some other conserved genes which are found in different groups of viruses, and this was studied by Natalia Yudin, Eugene Koonin, and Didier Raoult. And these elements encode such proteins as genome packaging ATPase or adenovirus like cysteine protease, and there is a specific relationship between polyntons and virophages, and also with bacterial viruses of the Tectiviridae family and adenovirus. So these transposons seem to be something in between viruses and transposons, because we have some viral genes and some transposon genes, but the capsid proteins could not be initially identified in polyntons, so still leaving them somewhere in between the two. And all these viruses to which polyntons are related, they have the same type of capsid protein. So these tectiviruses, adenoviruses, virophages, and this capsid protein is known as the double jelly roll capsid protein, shown here on the right. So it has two jelly roll domains within the same protein, and this is the major capsid protein forming the icosahedral capsids of these viruses. As you can see here, this protein trimerizes is forming these pseudo-hexagonal building blocks and constituting most of the particle protein content. However, the minor capsid protein is not less important for building the icosahedron in these viruses, and is conserved in all viruses which using this particular type of capsid protein. And this protein is known as penton protein. And more recently, we were able to identify homologs of both the penton protein and the major capsid protein in the polyntons. And specifically, the capsid protein of polyntons was most closely related to the capsid proteins of large double-stranded DNA viruses of the Phycodnaviridae family, such as Paramecium bursaria chlorella virus. And the conservation of the major capsid protein was closely followed by the conservation of other proteins predicted to be involved in the virion morphogenesis, specifically the minor capsid protein, the genome packaging ATPase, and the maturation cysteine protease. So all these genes are traveling together as a block in these polyntons. which suggests that perhaps under certain circumstances, pollutants are still capable of forming viral particles. And this realization that pollutants are even more virus-like than previously thought inspired us to look more into their relationships with other viral genomes, and that we did. And what we found is that the genome organization of different pollutants is quite diverse. So some pollutants display very similar genome organization to the bacterial tectivviruses and virophages, specifically MA-virus virophage. And however, other pollutants in the genome organization were more closely related to adenoviruses, as shown here. Then I mentioned that pollutants also share the polymerase gene with single-stranded bidnaviruses. And then also polymerase gene is shared with eukaryotic mitochondrial and cytoplasmic plasmids. And these cytoplasmic plasmids, interestingly, they encode the suite of proteins which allows them to perform transcription in the cytoplasm. And these genes display a specific evolutionary relationship to their counterparts in viruses of the order Megaviralis, including the giant mimiviruses. So these include mRNA capping enzyme, which has specific 3-domain organization, RNA polymerase, and also the D11-like helicase, which is important for cytoplasmic transcription. So, and then the transposase or integrase of pollutants, it's related to the transposase of ginger-1 transposons, which, in addition to the transposase domain, they encode the adenovirus cysteine protease domain. So again, two genes shared with the pollutants. And all these relationships between different elements, they can be displayed as a network where nodes are different elements and the edges are shared genes. And in such a network, pollutants come as a central hub in this network which share the largest number of connections between different elements. So to understand what all these gene relationships mean, we build an inclusive tree of protein prime family B DNA polymerases, where we included all known prokaryotic elements with protein primed DNA polymerases from bacteria and also from archaea, and then all known eukaryotic elements with this type of polymerase. And there are two take-home messages. First one is that all prokaryotic viruses form one clade, and then all eukaryotic elements with this polymerase, we form another clade. So that all eukaryotic elements share a more recent common ancestry. All elements of the genes of this gene shares more recent common ancestry with each other than with prokaryotic counterparts. And then the second important thing is that in the clade which included pollutants were at the base of the clade which contained different groups of viruses, specifically adenoviruses, bidnaviruses, and virophage, which is not shown here, and also cytoplasmic plasmids. So these different groups of viruses and plasmids seem to be derivatives from pollutants. And taking into consideration all this phylogenetic evidence from polymerases and all other evidence from comparative genomic analysis and structural comparison, led us to propose the following scenario for the origin of different groups of double-stranded DNA elements in eukaryotes. So what we believe is that pollutants are derived from bacterial tectivviruses, which could have been introduced into prokaryotic cell along with a bacterial symbiont, which eventually could have given rise to mitochondria. And then the major difference between tectivviruses and pollutants is that pollutants encode the cysteine protease and integrase. And both of these genes could have been introduced into pollutants in one shot by integration of a ginger-1-like transposon. And then domestication of this transposon would equip pollutants with the integrase and the protease. And then this was probably a game changer for these elements because it allowed them to lead a dual life cycle or lifestyle. So they were able to integrate with the aid of this interface into the genome of the eukaryotic host. And then it also different lineages of polyntons have diversified following very different trajectories. So, for example, in some elements, the morphogenesis unit, including capsid proteins and genome packaging ATPase, were lost completely, leaving them with only the transposon-like lifestyle. In other areas, the integrase was lost, like in the case of adenoviruses, and these viruses then can no longer integrate into the genome, but we can only lyse the host cell. Then recombination with parvovirus ancestor, as I mentioned, gave rise to bidnaviruses. And then the most exciting part of all these connections which we observed is the relationship or contribution of polyntons to the origin of cytoplasmic elements, the DNA elements in eukaryotes. And there are two groups of double-stranded DNA elements which are able to replicate in the cytoplasm of eukaryotes. And these are cytoplasmic plasmids and members of the Megaviralis order. So, and both of these groups of elements, they necessarily have to encode their own cytoplasmic transcription machinery, which includes the capping enzyme, RNA polymerase, and some other proteins. And what we propose that the escape from the nucleus happened once in the history of these elements, and then this escapee probably followed two alternative paths. One was to go through the reduction to become a cytoplasmic plasmid. And these elements, they lost all the capsid genes and they are restricted to vertical transmission within their host cells. And Megaviralis probably has followed the alternative pathway by increasing the complexity and to become more and more independent and autonomous for cytoplasmic replication. And this would necessarily require the replacement of a protein-primed polymerase with an RNA-primed polymerase, because protein-primed polymerases, they can support replication of only short, relatively short genomes of around 45 kb, and the replacement of this polymerase would allow megaviruses to expand the genomes almost indefinitely. And then the third way how an element can survive in the cytoplasm was probably followed by virophages, which instead of acquiring their own machineries for replication within the, or transcription within the cytoplasm, they learned how to hijack the transcription machinery of the larger siblings, specifically mimiviruses. So, and the natural question is how these small viruses or small capsids could could evolve into such huge capsids like in giant DNA viruses. And structural comparisons of different capsids from different viruses indicate that there are at least two pathways how size can be increased and allowing incorporation of bigger genomes. So one is followed by adenoviruses. So adenoviruses and tectiviruses, they have capsids which are identical in terms of number of capsid proteins, both major capsid proteins and minor capsid proteins. So they are indistinguishable. However, the capsids of adenoviruses are considerably bigger. And the trick which was done by adenoviruses is that the size of the capsomer, this hexagonal thing here, it increased in adenoviruses. So the two beta barrels in the capsid protein of adenoviruses are further away, which makes the hexagonal capsomer instead of 80 angstroms in PRD1 to become 97. And then the removal of the internal membrane allows adenovirus to pack a genome which is twice the size that of PRD1. And the other strategy, which was probably followed by NCLDVs or megavirales, was to increase the triangulation number of the capsid. And what this means is that they increase the number of building blocks they use from one five-fold vertex to the other. And this is, as you can appreciate here, the number of these capsomers in the two capsids is really different, which allows to build you very large triangular facets and large icosahedra. And what could be the molecular mechanism of that? And probably the X-ray structure of BRD1 might hold the answer to this question. So at least in BRD1, if we peel off the layer of the major capsid protein, which is shown here, underneath it is the framework of protein B30. And B30 dimers, they connect the five-fold vertices of the icosahedral capsid. And the protein was called tape measure protein, because it fixes the distance between the five folds of the capsid. So, and if you increase the size of this tape measure protein, you will have the five folds at increasing locations. So it's a kind of scalable system, which, or ruler, which allows you to build bigger icosahedral capsid. And this protein also is important for organizing the major capsid proteins within the icosahedron. As you can see here, one triangular plate is, of a major capsid protein is shown. So these kinds of two adaptations, increasing the building block and also increasing the number of building blocks within these viruses with double general capsid proteins, allow to reach a diversity of capsid sizes and genome sizes accordingly. So to conclude, there are all kinds of elements in the biosphere, viruses, plasmids, and different transposons, and all of these elements, they exchange genes quite frequently, and they in fact interconvert from being, for example, a virus on one day to being a plasmid on the other day, and then going to something else again, and this generates the diversity within the virus, which is quite intriguing to play a detective and trying to figure out how different groups of elements are related. And now is my kind of real conclusion slide. So I think this movie, it reflects very well what happens in the viral world. So it reminds me of a dance between different elements or different genes where different partners are exchanged. And as you can appreciate, this is not very well choreographed or not very kind of neat and quite chaotic. However, yet they dance. You know, and I think that is what happening in the viral world. And with this, I would like to thank all the people which I had the pleasure to work with over the years. So Patrick Porter and David Savishvili from our unit at Institute Pasteur, Eugene Kunin from NCBI, with whom I interact closely on different groups of viruses, and Simon Roux and François Hinault from Thermoferrand, and Jaina Ravanti and Denis Bamford, with whom I work on Gemini viruses and more general aspects of virus evolution and virus origin. And thank you all for your attention.
Well, it is interesting to see that your generation is born with the idea that everything is changing with everything, which is reassuring compared to the previous generation. So this is very reassuring. Well, as I told you when we were discussing, I think that it's going to be worse as the next step when we look at the recombination among genes. That will trouble even more than what it is. So the idea, the general idea is that we can base the phylogenetic analysis thinking that gene is a unit. I think is completely wrong. Gene is a sequence with a function.
Well, all this recombination, it is certainly true, and it is obvious that this is the case, and it takes place on all levels of genetic information. However, there are some restrictions also. There are some separation of different elements, and it very much depends on what kind of time frame you are interested in. For example, it doesn't seem to have occurred in these single-stranded DNA viruses with like Circoviridae nano and Gemini viruses. Of course, before that, before they became separate groups of viruses, there was probably a combination between them, and eventually their genes, they are recombinant at the beginning. But if we focus on the like particular groups, it doesn't seem to be going on, at least not on the same scale of evolutionary distances. So we don't exchange these genes with very distant things. And this speciation thing, of course, it's true, so it limits them, and it does not allow them to acquire very widely different things from things which have been evolved and adapted to different environments. So
did you look at if the divergence could be partly related to an exchange with the host? Recombination is part of the gene of the host, specifically. You know, if you select a sequence that diverge among your viruses, and just select this one, and compare that to all the genome of the host, and try to find out if you get signature showing that it is recombination and it's not evolution. Well, it's evolution in general, but it's not a step-by-step evolution.
yes, well, we haven't done that, and one of the reasons is that there are not that many hosts for these, like these eukaryotes and all these plants, for example, for Gemini and nanoviruses, we are not simply sequenced. So we don't have that data. But we actually haven't thought about that, and this hasn't been done.
I think because what is interesting, the reason probably why we can make a tree out of it is that because they don't have a lot of partners. So it's maybe it's quite easy when you get, we did that on Entrusted Ducks, for example. It's quite easy when you know the partner, and because there is not plenty of partner like outside, but when it's very, we call that allopathic, when it's allopathic, it's quite easy to look even at very, very small exchange, like say 5 or 10 bases, and it makes sense the way it's at diverge may not be just by rolling like that, just by exchanging sequences.
Well, I would say that these nanoviruses and Gemini viruses, they do co-infect the same host, so they do meet in eukaryotes, and yet there is no evidence that they recombine. So probably what nanoviruses have is no longer suitable for circoviruses, because there are not only the function of the gene, but also there are other elements within the gene which are important, the secondary structure of the genome in that particular location, which might be important for virion assembly. So there are multiple functions of one piece of nucleic acid. So probably these are already incompatible between lineages which have diverged to a certain threshold level.
I have one example. I'm working on antibiotic resistance in bacteria. There is some example and one main example of a recombination between two parts of sequence that are completely different to build the new sequence that is chimeric and that could give the new function. Did you have seen that between recombination between viruses and prokaryotes? It's a completely new function that was unknown before, just by a recombination event.
I cannot think of any example, so nothing pops out, so it's probably not as frequent. Probably in the evolution of viruses and all this recombination going on, probably many of viral genes are results of this kind of recombination between unrelated proteins which are gaining a new function. So I'm quite sure that this was the case with many viral genes, but nothing comes to mind which would happen now, kind of recently. Yes. Okay. But it's a general phenomenon to living organisms to do this kind of recombination.
And it probably happens more frequently in virus living in amoeba because, of course, there is plenty of opportunities to exchange genes compared to the others. So you can see much more exchanges and many sources of sequences in virus living in amoeba compared to the others. D'autres questions?
My question is that what is the biological advantage that such a mechanism of mosaicism puts on some of these viruses been able to discover, like between single-stranded RNA and that of the DNA? Is there any biological advantage?
So what I think about why they exchange with replication proteins so intensively is that like in these lineages of like established virus families, the capsid protein and the replication protein, they are already co-evolved, so they match each other very nicely. But when there is like dramatic recombination event happens, so you get the capsid protein from one virus which has never seen this replication protein, so the match is not exactly very good. So when there is a sampling, exchange and sampling, and the other recombinant is not so much less fit than the parental one, so that it can kind of exist and leave a trace. In case of those lineages which are already adapted with the capsid and replication protein, probably recombination does occur, but these variants which are emerging, they are not fit at all compared to the parental one, so they are just wiped out and we just don't see them. But in this case, when you exchange these genes from very distant parts of the virus, then this is the way how they try to find the best partner, and that's where the fitness comes from eventually. That it becomes more and more fit. We accumulate point mutations, we try different combinations. And because this event of these RNA and DNA virus recombination, it seems to be rather recent, because we still detected at the sequence level quite easily.
My question is regarding polymerase. So we have identified that like your copy numbers in eukaryotes. So what is the criteria you have made for that? Like, is there any GC variation between that region for the polymerase? You are saying that polymers are the repetitive elements kind of thing in eukaryotes. Is there any GC percent variation according to organisms?
Not exactly. So it doesn't seem to be a kind of co-speciation of these polyntones with the hosts. For example, if you do phylogeny of the polymerase or the capsid protein, so what you see is that there are clades and they do not coincide with the host. For example, from fish you have polynton in one clade and in the other, and then in the same clade you have something from Drosophila and from alligator. So there seems to be kind of either this divergence of these elements has occurred before these elements, like these cellular organisms before the diversification, or these polyntons from different clades were introduced later into the genome after the diversification of these organisms. So there is no strict correlation kind of according to the host.
Ok, merci.